Hello, everyone. My name is Xin Xie. I'm working as a research scientist in New York University, Abu Dhabi. Today, I'm going to share with you our ultrasensitive detection method for SARS-CoV-2 we implemented in NYUAD. One of the main challenges in COVID-19 screening is the high false negative rate of a standard qPCR detection method, which is estimated from 10 to 20% false negative rate. As many of the infected people are asymptomatic carriers, many of them may have very low in their samples, which pose a major problem in efficiently detecting those samples. So to cope with this problem, we aim to implement a cost-effective, sensitive, and scalable method for monitoring COVID-19 in our community. We adopted the Microfluidic qPCR system for two main reasons. First, it can reduce the sample and reagent requirement. Secondly, it is highly multiplexable for analyzing a different number of the assays. For example, in the 192 and 24 chip, we can analyze 192 samples against 24 different assays at the same time. So first, we aim to determine the sensitivity of our method. We used a Twist RNA, which is a synthetic RNA covering the 99% of the SARS-CoV-2 genome. We also included a plasmid positive control, which contained the DNA sequence of the nucleocapsid gene of the virus. We compared two different workflows. One is with the preamplification step before qPCR, because preamplification has been previously used in studies involving limited amount of the genetic materials. We reason the inclusion of the preamplification step will enhance our detection sensitivity. So using a series dilution of the Twist RNA and the plasmid control, we clearly show here for both Twist RNA and the plasmid control, in the presence of a preamplification step, our methods are able to detect the viral genetic material at 1 copy per microliter range, which clearly increased the sensitivity compared to the method without preamplification. This is increased by at least 1,000 times. And next, we aim to validate our method using clinical samples. We collected 90 positive diagnosed samples and 91 negative diagnosed samples from accredited diagnostic lab in Abu Dhabi regions. And we analyzed in our Microfluidic qPCR platform. So we compared our detection results with a clinical diagnosis. We have to remove a few samples due to the inconsistency among the replicates because the bubbling issue in the manual loading. So in our the results comparison shows we can detect most of the positive samples. However, in the negative samples, our method also detects around 90% of them are also containing viral genetic materials, which means these are likely to be false negative samples. And when we estimate the viral load of these samples using the standard plasmid controls, we clearly see that this potential the false negative samples contains very low viral load, around 1 copy per microliters. So, for those negative samples, their RP have a good CT value, which means the quality of the samples and the ion extraction are good enough for the viral detection. So therefore, we conclude that our method has enhanced the detection sensitivity compared to the standard RT-qPCR. Now we have improved our workflow by including the automation. In this video, you can see like we are using the liquid handler to automatically load the sample into the microfluidic chip to to avoid any inconsistency during the manual loading. We also build a database to record every step of our workflow. And finally, we have implemented a QC step and algorithm to automatically report the sample status and the quality of each run to enhance our throughput. And this has been routinely used in our screening activity in NYUA. Lastly, I would like to thank for all the people involved in this work, especially the researchers in the NYUAD COVID-19 response task force. And I will thank you for your attention.